Hello everyone, my name is Sreekar Edula, and this is my study of MRSA. So MRSA is one of the most common skin infections caused by bacteria, and methicillin-resistant Staphylococcus aureus poses a significant threat in the healthcare setting worldwide. And this presentation aims to delve into the complexities of MRSA infection, and I want to mainly focus on the USA 300 strain, which is a specific type of the infection, and I want to understand the origins, transmission routes, and the resistance mechanisms of MRSA, and why it's crucial for effective prevention and management strategies in both both community and hospital-acquired setting. So MRSA, we need to classify the different types of strains, and commonly they're classified between the hospital-acquired and community-acquired infection. The hospital-acquired infection is typically found in patients in healthcare facilities, while the community-acquired infection spreads within community settings, such as sports games or public gatherings. And recognizing these distinctions is vital for implementing targeted control measures and minimizing transmission. The USA 300 MRSA strain has a fascinating evolutionary history, and it's very long. So it's originated from ST8 MSSA in Central Europe and later spread globally, and its emergence as a predominant community-associated strain in the United States underscores the importance of its understanding its genetic makeup and valence factors for effective disease management. So there's two different mechanisms for why MRSA is resistant to methicillin and different types of antibiotics, and this includes two different parts: the staphylococcal cassette chromosome, the SCC MEC element, and the MECA gene. And these two genetic components alter the bacteria's cell wall structure, the peptidoglycan cell wall structure, and this renders the traditional antibiotic ineffective. And understanding these mechanisms is crucial for developing novel treatment strategies against MRSA infections. So the image on the left shows how the MECA gene works its way into MRSA and makes it methicillin resistant, while the right shows the different types of elements of the SCC MEC component chromosome. So the different types of serotypes, which are designated based on these combinations, these five different components of the SCC MEC. So USA 300 virulence factors. The USA 300 MRSA strain is renowned for its virulence attribute to a plethora of toxins and proteins it produces. These virulence factors enable USA 300 to evade the host immune system, invade tissues, and cause severe infections such as necrotizing pneumonia and bloodstream infections. Understanding these virulence mechanisms is essential for devising targeted therapies. So MRSA causes multiple different types of pathological complications, and on the left we can see a few of them, including pneumonia, dermatitis, spinal osteomyelitis, central line associated bloodstream infection, acute respiratory infection, etc. And these MRSA infections can lead to all of these, and you can see that these different complications range from simple skin and soft tissue infections all the way to life-threatening conditions like septic shock and pneumonia. And there's different factors that contribute to the severity and complexity of these complications, including immunocompromised states, the invasive procedure, and the antibiotic use contribute antibiotic use in the cure for these complications, and this necessitated prompt and appropriate medical intervention. So I ran over a bunch of case studies while I was doing my studies, and in this chart I display the information of geographical area, time period, sample size, strain, pathological complication, and the PMID, which is the source. So this slide provides an overview of the key case studies examining the prevalence and impact of USA 300 MRSA strain in different geographical regions, and these studies shed light on the diverse manifestations and epidemiological patterns of MRSA infections and emphasizes the tools needed for comprehensive surveillance and control measures. So data from the various studies highlights the prevalence of the USA 300 strain in both CA and hospital-acquired MRSA infections, and understanding the distribution and epidemiology of USA 300 is crucial for tailoring prevention and treatment strategies to effectively mitigate its impact on public health. So preventing MRSA infections requires a multifaceted approach, including stringent infection control measures and prudent antibiotic use, and the development of novel therapeutics, implementing robust surveillance systems, and fostering interdisciplinary collaboration. These are all essential for curbing the spread of MRSA and reducing its associated morbidity and mortality rates. So in conclusion, MRSA infections, particularly the ones caused by the USA 300 strain, can cause significant challenges to public health and cause a lot of harm. And you must understand the genetic and epidemiological and clinical aspects of this disease, of this infection, and then we can develop a targeted intervention to mitigate its impact and safeguard patient health. So still it's important we must continue to research and vigilance, and it's paramount in our ongoing efforts to combat MRSA infections effectively. Thank you.